It's worthwhile reviewing our method of mapping characters onto a phylogeny. This is something that's critical for establishing the score for parsimony analysis, and it also tells us about the history of evolution of a specific character. Now, for simple distributions of character states, that is, if we don't have a very, very complex one, it's rather straightforward. So for this exercise, what I've done is I've duplicated a phylogeny. So this is the exact same phylogeny 4 times. And the only reason we've done that is so that we have extra room to work and we don't get too cluttered and messy. So we are going to map this character onto this phylogeny. Now realize if we're doing an analysis, we would be mapping characters. Sorry, we have 6 characters here that I've labeled. We'd be mapping 6 characters onto different phylogenies to figure out which one is the best. And so that would be the process that would be involved in finding the best tree overall. But I just want to make sure that you know and are able to accurately map characters onto a phylogeny. On an exam, you might see a fairly simple set of characters that you have to map onto 2 or 3 different phylogenies to determine which one is the best. And we can review that if you need to do it. But this exercise is just to make sure you understand how to map characters. So I have a matrix here, a DNA alignment, and in reality we'd probably see a much, much longer alignment with most of the characters, or maybe many of the characters, being identical for all species and not being helpful or informative for us. But I've just kind of synthesized this down into 6 specific characters. Remember, each column represents a different homology statement. That's a character that we're comparing across species. For the first 3, they're rather simple and straightforward, and we don't need to do a Fitch optimization, although you could if you wanted. For the last 3, they're much more complex, and so we'll actually go into a formal Fitch optimization for characters 4, 5, and 6. Now, if you want to test yourself, it might be a good idea to pause the video and try to map characters 1, 2, and 3 onto this first phylogeny. And you can make marks across the lines and put a little number by them to show the characters. If you're struggling still, that's fine. Then start the video back up and watch how I do characters 1, 2, and 3. All right, so here we go. We'll look at character 1 first. Character 1 has species A, B, and C sharing a character state. They all have guanines, and all the other species have an adenine. So what we're saying is A, B, and C are different from all the rest. Now on our phylogeny, this is rather straightforward because A, B, and C are all in a single monophyletic group. We can make a mark across the ancestor to that group. We'll put a little 1 to indicate that that's character number 1. And that is the simplest, most parsimonious way to map a character that A, B, and C is different from everything else. So again, that's an easy one, fairly straightforward. If A, B, and C were separated from each other, then it'd be a little bit more complex. In other phylogenies, that might be the case, but in this one, it's easy. Okay? Character number 2. This one's a little bit more complex, but we can still do it with a little bit of thought and without a formal optimization. So species A, B, C, and the outgroup are all different from the other ones. So notice that A, B, and C and the outgroup are separated by that branch from all the other species. So I can put a mark across that branch. We'll put a little number 2 there representing that that is a mutation. Now, the remaining species are not completely uniform. Species D is different from the other 3. And there are 2 both equally acceptable ways to do this. And without further taxon sampling, adding extra species that would be intermediate here, we don't know which is the best, and either one works. So either this little mark that we've made here on this branch represents a mutation from the ancestral state of a thymine into a guanine. And if that's the case, if that's a guanine, then we would need to put a separate mark here representing that transition to an adenine in species, the ancestor of species E, F, and G. And so that would explain the distribution. Now, alternatively, and we don't know again without some additional taxon sampling, but if that represents a mutation into an adenine, then we would make a mark across that branch and put a little 2 by it to represent a mutation in that character to a guanine. And so either one works. For as far as evaluating which trees are better, it doesn't matter whether you put it here or here. You've still only got 2 marks. And so we've done it in the simplest way possible. So we've completed a mapping of character number 2, and you can again put it either on that branch or that branch it means something slightly different as far as the character states and when they evolved. But as far as evaluating the length of the tree, it doesn't matter. You just have to do one of the simplest ways. Now, again, remember, you could come up with all sorts of conspiracy theories saying it was gained and then lost and gained and lost. And you can make really, really complicated scenarios and still come up with the same end character states for each of these species. But we're trying to find the simplest, or at least one of the simplest ways that we can map that. So we're done with character number 2. Now, character number 3 is also a little bit more complex, but not too bad. It says that species A and C have a thymine. Everything else has a guanine. And so A and C are different from everything else. Now, because A and C are not their own closest relatives, we cannot put a single mark in the ancestor of A and C and be done. It's a little bit more complicated. So for character 3, we have essentially 2 options. And again, either one works as far as determining the length of the tree. They're equivalent. They say something different about the evolution of the ancestor, and we couldn't pick one over the other unless we had some additional species represented. Maybe they're close relatives of these we could include, see what their character states are. That might shed some light on it. But for our purposes, we could do either one. So either A and C are the same because of convergence, right, which I could mark like that, put little marks across. We'll put number 3s just so we don't get confused with character 1 and 2. And A and C are the same because there was a convergent mutation in the ancestors of those species. Or an alternative is we could have had a mutation in the ancestor of all 3 of these, put a little 3 there, and that would represent a mutation to a thymine. But then B had what we would call a back mutation, where that ancestral thymine turns back into a guanine. And from a strict parsimony interpretation, it doesn't matter whether we mark it this way: a gain of a trait, loss in one of the descendants, that's a simplesiomorphy, or if we mark it as a simple convergence. Either one works from a very strict parsimony framework. Now, we raised the idea, if you'll remember, of, well, what if all of our mutations are not considered of equal weight or equal value? Then perhaps we would prefer either a convergence or simplesiomorphy. One might be a better one if we gave different weights. But from a strict parsimony framework where everything is treated as equivalently the same, it doesn't matter which you mark. So we'll leave this as a convergence, but if you put it as a simplesiomorphy gained here and then switched back in species B, that works fine. Either one explains why A and C are different from all of the others. Okay? So we've mapped characters 1, 2, and 3 with a little bit of complexity for some of those and some explanation of what's going on as we map them. For the remaining 3 characters, if you look at them and if you try to do them just kind of by thinking about it, you're probably going to struggle. If you would like to and like to really give yourself a challenge, go ahead and do it. But I'm going to show you a way that will work for any character. If it's a simple character like 1, you could do Fitch optimization, which is the method I'm going to demonstrate, but it's probably a little bit of a waste of effort. But any time you're in doubt, this method is a way to guarantee that you find one of the most optimal mappings. Sometimes, as we demonstrated with character 3 and character 2, there's more than one optimal mapping. But we want a little kind of step-by-step method to guarantee that we can find the best way to map characters onto a phylogeny, no matter how complex they are. Okay, so let's do character number 4. So I'm going to put a big 4 above this tree, and just because it gets kind of busy and messy, we're going to use a separate tree. These are all identical, but we'll use a separate tree for mapping characters 4, 5, and 6 just to demonstrate this. So this is Fitch optimization. Now, to begin with, what we do is we're going to write the character states above each species just to help us remind us, and we don't have to keep going back and forth to the matrix. So for character 4, outgroup and A and B all have guanines. So we'll put little guanines above each of those. My handwriting's not real great when I'm using a mouse. For character 4, C and D have adenines. So we'll put little A's above those. Okay. And then we've got, this is a complicated one, right? E and F have cytosines. And species G has guanine. So you look at it, and it's like, wow, that's a really complicated character. And some characters are like that. So this is the process of Fitch optimization. There are two parts to this. The first one we're going to do is called the down pass. Again, sorry for my handwriting, but let's go through and do a down pass. What we do with the down pass is we begin at the tips and we work our way down to the more ancient ancestors. And we assign ancestral states. So let's start with E and F because that's an easy one. E and F both have a cytosine at that position. So we are going to assume the ancestor had a cytosine and we'll assign a C to that node. Easy, right? Now we're not going to map any characters until we've assigned all the ancestral states. Now notice that if we go down now to the next deeper node, the ancestor of this E and F node and the species G, we have a cytosine here. We're only going to look at the most closest ancestor, which is that node right there, and we've assigned a cytosine, and then a guanine. So we don't know. We can do either a C or a G. For the down pass, we don't know because we've got one C from above here and one G, like they're voting, so we'll throw them both in there. We go down to the next deepest node, and notice that there is a D there. So now suddenly, or sorry, that's species D, there's an adenine there. Suddenly now we have a vote for C, a vote for G, and again, I'm taking that from this node right above it. And now from this other branch up here, I have a vote for A. So I could have all of them, and we won't decide until later which it is. Okay? So far, so good? So now we would go down to the next deepest node, but I can't do that node yet because I haven't decided what these nodes above it are. So let's go up here and decide what these nodes above it are. Let's do B and C, and we need to put a G and an A because B has guanine, C has adenine. So I'm going to put both. Let's make sure that looks like a G and not a C. And then we'll go down to the next deeper node here, this node. Now, when I'm assigning that one, I have a vote for guanine and a vote for adenine from the node above, and I've got a second vote for guanine from that branch right above there. So 2 guanines, 1 adenine, and this one we have plurality or majority rules, whichever one has the most votes wins. So I would put a guanine, and now I have established that ancestral state. Now, this one's still ambiguous. These two are ambiguous. We'll show you how we resolve that ambiguity later. We're going to do that with what we call the up pass. So I've assigned a guanine there. So now that I've assigned that node right above, I can come down to this deeper node. I've got a vote for guanine, and from this node I've got 3 votes, but one of them is guanine. So 2 out of 4 are guanine, so I can assign that simply as a guanine for the ancestral state. Right? 2 votes for guanine, a vote for cytosine, a vote for adenine, and suddenly we've got that one resolved. And of course, now we can do the very most basal one. This ancestral state is also a G. That's easy because this G is above. We assigned a G on that node. And that's the down pass. So during the down pass, we assign either a set ancestral state for each node, or we say, okay, I'm not sure, it's got some ambiguity, and we would assign multiple states that we don't know yet. Now, this next part of the Fitch optimization is called the up pass. And we're going to go in reverse. We're going to, instead of working our way from the tips down to the nodes, we're going to work our way from the most basal nodes, the most ancestral nodes, all the way up to the tips. Now, if in our down pass we assigned a set ancestral state, we're not going to mess with it. We're not going to change it. But if the ancestral state is ambiguous, we're going to use information from below to help us decide what the ancestral state should be for the ambiguous nodes above it. So G and G are set. Those are easy. But as I come up here now, oh, notice I've got an ambiguous one. But because the one beneath it is a G, I am going to assign this node to be a G. And I use the information below it to decide which of the 3 it should be. Okay? And again, now that this is assigned to be a G, it is set. I can use that to help me establish what I should assign this one. And so we will also assign that one to be a G, again, using this one we set down here to be a G. Sorry, my G's look a little bit like 6s, but a G, and now I have assigned ancestral states to all of these nodes. Let's go and do this last ambiguous one. Because it's a G there, we don't have enough space there, so we'll write my arrow coming up there. I can assign that one to be a G. Again, using the information from the node beneath it to help resolve that ambiguity. Now that I have all of my ancestral states set, the last thing is just to mark changes. So notice ancestor is a G, that's still a G, that's still a G, that's still a G. So it's not until we get from here to these ancestors that I have a change. So I need to mark a evolutionary event across that node, right? We could put a little 4 by it, but we know this whole tree is character 4. So the ancestor here was a G, that ancestor is a C, so I have to mark a change on that node. This ancestor is a G, but that species has an A, so somewhere along there was another mutation. And then finally, we have a third mutation here. Ancestor is a G, but species C has an adenine there. And so we mark a third mutation. So this is the most parsimonious way, 3 changes to map for character number 4. Okay, we're going to practice. I know that's kind of complex, but we're going to practice with these other trees down here for character 5 and 6. Now, to do that, let me just kind of move things around. We're going to use the exact same process. Now, if you feel comfortable and you're not totally confused at this point, we'll leave those there so you can look at that. Maybe we should also move character, well, we can do, yeah, let's just move this one down. So this is character 4 there. I'll move these just so we remember what we're doing, down pass and up pass. And if you feel comfortable and confident, you're not too lost, maybe go ahead and try to do that on your own. Do this one for character number 5. Use that tree, and then we'll use this tree for character number 6. So try it if you feel confident. If not, follow along. And I'll maybe speed up a little bit, but I'm going to do this exact same process that I've already done. Okay. So again, to keep track, first thing we're going to do for character 5 is write all of the states for each of the species. So species A has an adenine, species B has a cytosine, species C has a cytosine, species D has a thymine. And again, I'm just going down the column here and writing in what it is, so I don't have to keep looking back and get lost or confused. G, G, and T. So I've written all of my character states for character number 5 for each of these 8 species. Okay, so now let's do our down pass. So we'll start with E and F. That's easy. They're both G's, so we'll assign a G right there. I can't decide here because the node right above it is a G, and then this branch right above it, the species, has a T. Don't get your species designation letters confused with the A's, T's, C's, and G's for the nucleotides. Okay, I go down one more. Now notice species D has a T, and this is a G or a T. So I'm just going to assign that a T because I have 1 vote for T from the branch above it, and a vote for T and G from the node right above it. And so there we go, it's a T. I can't do this next node yet because I have yet to resolve these. So B and C both have C's. I'll put a C at that node. And then I can't decide here because there's a C above it and an A for species A, and adenine for species A. Let me just make this a little smaller so I don't get confused which is which. And this is either an A or a C. I don't know which one. So we'll leave it ambiguous right now. Now notice when I come down here, I've got a vote for A, a vote for C, and then the node right above it here is a vote for T. So I have to put all of the above because I have 1 vote for each. No one has a plurality. By the way, you know, if you don't know the term plurality, it's when you have the most votes, but not more than 50%, right? So if there's more than, so for instance, if you had an election and there were 4 candidates and one candidate got like 40% of the votes and then the other 3 candidates split the rest among them, but no one had more than 40%, that would be a plurality. And in elections, depending on the rule, sometimes you win with a plurality. Other times you have to then go back in and either revote or have the 2 highest people go in so someone has a majority. Anyway, but same thing, when we're mapping like this, a plurality is good enough. So if I had, say, 2 out of 5 characters, well, there are only 4 possible characters, so we would at least have 50%, which is, I guess, technically a majority. So anyway, A and C, T, everyone gets 1 vote, so we have to put all of them there. Now, when we go down to our most basal ancestor, that node right there, we have a vote for a T from the outgroup, an A, a C, and a T here. And so 2 out of 4, boom, we assign that a T. We're done with our down pass. So now we start down here and we use the information that we've figured out to do our up pass. T, changes this ambiguity into a T. Once we've established that that's a T, this is where it gets a little bit tricky, but you can do an either or here. So notice this is a T down here, and that T can't help me decide if this should be an A or a C. And so when you can't decide if it's an A or a C, it's your choice. It doesn't matter. So we'll pick this into be a C. Okay. If I assigned it with an A, it would have the exact same number of marks. We just have to put them in a slightly different place on the tree. Now this T helps us to decide that the one above it should be a T. Right? And so now we've assigned all of our ancestral nodes. We can map traits on there. A T, there's a T there, there's a T there. So we don't need to make any changes along here because all the ancestors are a T. We need to make a change somewhere along here. And so we change that T so that ancestor can be a C. And then we have to put one more mark here so we can have that mutation being an A. And all we do is look at what the ancestors are. If there's something different above it, we need to mark the mutation on the branch. Okay, so here we go. We'd mark it right there to say that this ancestor, which was a T, turned into a G. 1, 2, 3 marks, and I'm done. Now, just so you are clear, you could do it this way also. If we decided that this ancestor should be an A, and again, you could do either or because the node below us is assigned a T and doesn't help us decide. This one would still be the same as we had it before. We'd still assign that one a T because of the node below it. But here, if we decided we wanted an A, we would still make a mark here to represent this ancestral T changing to an A by the time we get to there. But then we would put the mutation from an A to a C right there. But still, 3 marks, put my mark back here. 1, 2, 3, either one works fine. Okay, so hopefully you're starting to get, I know this is one of the more complex things we do, but let's go ahead and do character number 6. Okay, I'll speed up just a little bit. So again, I'm just going to write my character states here. CC, species C has an adenine. Species D has a guanine. And then we have a C, C, T. Okay, down pass. I'm going to assign either a set state or an ambiguous state to each of these nodes as I go down. Okay, C or A for that node. Yeah, let's make it a little smaller. It's kind of crowded in there. And then this one gets assigned a C because we have a vote for a C here, a vote for an A, and then a vote for a C from that species. So now we've got C, CGT. That's easy. We assign that a C. The ancestor, or the outgroup there is a C also, so we assign that ancestor a C also. We now do our up pass, and we can use information from the things below it to assign states. A little arrow meaning I'm going to change that one into a C. Now that that's a C, it helps us establish the one above it should be a C also. And now all I have to do is map where they're different. If this ancestor is a C, we have to have a mutation right there because that's a T. If this ancestor is a C right here, we have to have a mutation along that branch because that's a G. C, C, C all the way up here, C. The only other mutation we have is right there. And so now I've got 1, 2, 3 mutations. That's the simplest way to map that character onto that phylogeny. Okay? Now, hopefully you realize amongst all this complexity, and of course with the simple mappings also, we are making inferences about the ancestors. So I could now, if I wanted to, for any one of these ancestors, let's say I pick that ancestor, and I want to say, well, what was its DNA sequence like? What's the most likely sequence for that ancestor? Well, for character number 1, because I've mapped the change there to a guanine, it must have an A for position number 1. Character number 2, notice I mapped a change there, which for character number 2 is a change from a thymine to a guanine, and then I mapped a change to an adenine there. So that ancestor, based on the mappings for character number 2, has a guanine. For character number 3, since all the changes were up here, that's easy. It's just another guanine. For character number 4, we've already mapped character number 4 out here, and so for character number 4, that ancestor, we assigned a G. For character number 5, we assigned that ancestral state to be a T. And here, after the down pass and the up pass, we assigned it to be a C. So not only are we figuring out which set of relationships are best based on this parsimony criteria, by mapping characters onto this, we're explicitly estimating, hypothesizing, what the ancestral state was. So I could say, based on these modern species and their states, that the ancestor of this had an AGGGTC. Now notice that's kind of cool. It's different than any other living species, right? But I've been able to reconstruct an ancestral state, and I could do that for any one of these ancestors if I wanted to. And so in theory, you could even reconstruct, say, ancestral protein sequences, and if it was different than any other protein in an extant living species, it would still be our best hypothesis of what that protein was. We could synthesize it artificially. We have the capability to do that now and see if it was functionally different than any of the other proteins. So it's kind of a cool process where we could get some information about missing ancestral genes, not missing, but now extinct ancestral genes, and see if they were actually functional. So there are some really cool applications to this, in addition to figuring out is phylogeny A better than phylogeny B.